Welcome to The Explainer. Okay, today we're diving into an absolutely wild biological mystery. We're looking at how your adaptive immune system prepares for threats it has literally never encountered before. Think about it. Throughout our lives, we're bombarded by countless microbes, right? And a lot of them are mutating at breakneck speed, changing their structure daily. Or maybe they haven't even existed in human history until this exact second. So how on earth does your body gear up to fight an enemy it knows absolutely nothing about? It's just a fascinating puzzle.
I mean, it really is the ultimate impossible puzzle. It's like a locksmith paradox. How does your body forge a perfectly shaped key for a microscopic lock that doesn't even exist yet? Because, well, the adaptive immune system faces one massive fundamental limitation. It can't predict the future. It has zero way of knowing what specific antigens or crazy mutated viral strains are going to show up tomorrow or next week or, 10 years from now.
So let's just bust a common myth right out of the gate here. Your body does not custom design a weapon after an infection starts. A new virus doesn't just hand over a blueprint for your immune cells to sit down and build a custom receptor from scratch. No way. Instead, millions of random weapons are already waiting in your body for a virus to accidentally select its own destruction. The strategy is breathtakingly simple, but oh man, is it powerful. Rather than trying to predict the exact future, the body just goes ahead and creates a staggering variety of possible recognitions way in advance.
Which brings us to part one, the ultimate immune paradox, preparing for the unknown. To actually solve this paradox, your body leans entirely on generating just a staggering amount of variety, a vital capability we call diversity. You gotta stop thinking of your immune system as a single super soldier that somehow knows every martial art. Instead, picture it as a vast pre-stocked library. We call this the lymphocyte repertoire. This repertoire is the complete collection of all those diverse antigen receptors spread across all your B and T cells. The true power of adaptive immunity? It doesn't come from having one perfect, all-knowing receptor. Not at all. It comes from having this overwhelmingly massive, diverse population of cells just waiting in the wings. But here's the catch. Inside this massive library, each individual mature lymphocyte only reads one specific book. By that I mean, it has exactly one main antigen receptor specificity. There's no single B cell out there carrying hundreds of different receptors for hundreds of different diseases. Nope, they are highly specialized, absolutely dedicated single taskers. So the incredible diversity of the adaptive immune system, it doesn't live inside one multitasking cell. Instead, it's spread out across this giant collective population of those specialized cells. Lymphocyte 1 perfectly recognizes target A. Lymphocyte 2 perfectly recognizes target B, and so on and so forth. Your immune system isn't a single master key trying to force its way into every door. It is a massive warehouse holding literally millions of uniquely shaped keys, anticipating millions of different potential locks. Moving on to part 2, diversity before specificity, the foundation of precision. Now, while diversity and specificity might sound like total opposites, having a diverse arsenal is actually the exact thing that makes pinpoint precision possible in the first place. You literally cannot selectively target a specific threat if you don't first have a huge array of unique options to choose from. Chronologically, it plays out like this. First, the body forms that diverse repertoire, creating that massive possibility space. Only later does an antigen actually enter to select and expand its matching clone. The antigen isn't the designer here, it's just the selector. It finds the one cell out of millions that happens to match it just by sheer chemical geometry, and it triggers that specific cell to multiply into an army. All right, part 3, the genetic mix and match. Infinite keys, limited genes. So here is the next major piece of the puzzle. If you need millions of unique keys, how do you actually build them without completely running out of DNA space, right? If the body needed a separate, dedicated gene for every single possible receptor, the human genome would have to be impossibly huge. We just do not have the space for that. So the body uses this genuinely brilliant process called VDJ recombination. That stands for variable, diversity, and joining. Think of it like a genetic slot machine. During the development of B and T cells, these tiny variable, diversity, and joining genetic fragments are mixed, matched, and rearranged. It shuffles a tiny number of gene segments into an astronomical number of combinations. And it does all this long before any pathogen ever even thinks about entering the body. So rather than storing one dedicated gene per receptor, the immune system economically stores limited genetic fragments just to generate a massive potential diversity. It's basically the ultimate biological life hack. You take a really small set of building blocks, and by combining them in virtually infinite ways, you generate a massive defensive arsenal out of a tiny genetic footprint. It is wildly efficient. Next up, part 4, the cost of diversity, time limits and friendly fire. Okay, so maintaining this massive library of biological keys, it comes with some severe physical limitations and, frankly, pretty dangerous evolutionary trade-offs. You can't have everything instantly, and having this much variety introduces some very real risks to the body. Because we only have so much physical space in our bodies, packing in millions of different specificities means we only get a tiny, tiny number of cells for each specific threat. So before an infection hits, the strategy is basically many specificities but very few cells per specificity. And the catch here? This results in a delayed immune response when a new virus hits. The body literally needs time to hunt down that one specific cell and multiply it into an effective fighting force. And this fundamental tension of the immune system is just fascinating to me. By randomly generating millions of receptors to cover all future threats, the body inevitably creates cells that accidentally want to attack our own healthy tissues because the generation process is, random, and it obviously doesn't know what a future virus looks like. It occasionally builds a key that accidentally fits our own biological locks, what we call self-antigens. It's an unavoidable side effect of doing this kind of massive random generation. So to survive this potential friendly fire, our body absolutely requires a process called immunologic tolerance to weed out or silence those dangerous keys that fit our own internal locks. A healthy immune system has to constantly walk this incredibly dangerous tightrope, maintaining enough diversity to fight off unknown invaders while strictly regulating and destroying the cells that would cause an autoimmune disaster. Without this internal quality control, the very system designed to protect us would literally destroy us. Which takes us to part 5, your unique immune map, a living repertoire. When you combine all these factors together, you start to realize that the theoretical math of the immune system and the actual physical reality inside you right now, well, they're two completely different things. The immune system is incredibly vast, yes, but it is not infinite. We really have to distinguish between potential diversity, which is virtually infinite based on all those math combinations, and your realized repertoire, which is restricted by your physical size, failed cell developments, and, of course, those self-reactive cells your body had to destroy. Not every theoretical receptor combination exists inside you at this exact moment. You only hold what you physically have space for and what has safely passed your body's rigorous internal testing. To make sure we don't end up with massive blind spots in that limited realized repertoire, our receptors rely on something called cross-reactivity. This allows one single T cell or B cell to loosely recognize multiple similar threats. Think about it. If every receptor only recognized one absolute mathematically perfect molecular shape, we'd never have enough cells to cover all the gaps. Cross-reactivity provides this beautiful biological buffer. It extends our defensive shield just enough to keep us safe even when we don't have the absolute perfect match. And because your repertoire is shaped by random genetic shuffling, rigorous selection, and your personal lifetime of infections, your immune map is entirely 100% unique to you. In fact, even identical twins do not share the exact same set of lymphocytes. I mean, that is just a staggering thought, isn't it? Your repertoire is a living, breathing record of your life. It's shaped by probability, your age, the vaccines you've had, and literally every cold or flu you've ever fought off. So the entire strategy of the adaptive immune system really comes down to this one phrase: generate broadly, select specifically. It's not just some cool evolutionary feature, it is the absolute prerequisite for surviving in a world full of unknown, constantly mutating threats. You cast the absolute widest net possible in advance, and then you pluck out the exact weapon needed right at the moment of infection. Which leaves us with one final thrilling mystery to uncover in our next explainer. When that deadly virus finally arrives and the clock is ticking, how does the body instantly search this massive million-strong library to find and multiply the one specific cell that can save your life? How does it actually initiate that critical clonal selection when every single second counts? Well, we're going to unravel exactly how that life-saving search engine works next time. Thanks for joining me on this explainer, and keep questioning the amazing world inside you.